So Joel told me to not just dive into details, but try to give a little bit broader view on how we think about physics and biology and how we go about building models in biology. So, and so I'll have sort of more of a philosophical, digressions in my talk than I normally would have. Forgive me for it. And I'll start with a bit of that philosophy. So where does physics fit into biology? Well, some of it these days requires no explanation whatsoever. There is sort of biophysics in the strict sense, which is relevant to biology. But, and I won't really dwell on that so much, but rather focus on so the situations where we borrow concepts liberally from physics. And so concepts and methods, for example, sort of attitude towards phenomenological modeling, which is very much favored in the world of theoretical physics and very much looked down upon in the biology world, but we would like to change that. And last but not least, I'll be emphasizing bits and pieces of where thinking about biology is actually enlarging the domain of physics. And perhaps thinking about biology as a physicist might motivate us to ask some questions about biology that we haven't thought of before. Well, so an example of this biophysics in the strictest sense, of course, is liquid-liquid phase separation, where essentially physics of mixing and demixing is driving formation of intracellular aggregates that underlie signaling and biochemical processes. And you all know about it. And Cliff Brangwyn and Tony Hyman might actually be collecting their prize in California, this week, this month. This is having tremendous impact in biology right now. But instead of focusing on that, let me give you a very low-key example of what I would call sort of off-label use of physics concepts. So if you look at the back of the hand, you will observe that the little hairs there are mostly pointing in one direction. They're polarized. So there's really a sense in which there is this collective order in polarization, the planar cell polarity, and physicists thinking about it immediately starts thinking about magnetism, because that's where the concepts of this collective order have been developed. But and these concepts are very, very relevant in the context of planar cell polarity, yet there is no magnetism. There is no magnetism involved in planar cell polarity. So ideas can really go far. And then, of course, another thing is there's no theoretical physicist, I think, ever talking about morphogenesis without paying homage to Darcy Thompson. And in my case, I just visited Dundee a couple of weeks ago for the first time, so very fresh in my mind. But in any case, Darcy Thompson, thinking about sort of geometry of living forms, thought of it as basically a mathematics problem and a physics problem, and gave us all this great cover for considering this as physics. But he, of course, was thinking about morphogenesis long before the advent of molecular genetics. And we have learned a great deal since then, obviously. So for example, we know that inappropriate expression of a single gene, antennapedia, in the head of a fly, will transform an antenna into a leg. So we know a gene which switches developmental program from leading to an antenna to leading to a leg, but that still leaves us with the question of what exactly is the difference between antenna and the leg, and how do you produce these different structures, different morphologies, just downstream of one program. So, and of course, distinguishing just between the antenna here and the leg, so that's ultimately quantitative phenotype. Distinguishing between the geometries of these structures is a quantitative question. And my battery is running low. My battery is full. It must be some other battery that's running low. Okay. All right. So wouldn't it be fun to actually revisit Darcy Thompson's agenda with all that we've learned from molecular genetics and ask a question of how genes actually do encode geometry? And we are here in a plant biology lab, and there is a striking example of a gene controlling geometry that was discovered in studying plants. Particular, so this one particular gene called SYN acts as a repressor of growth and transforms the margin of a leaf into this curly lettuce type. And we understand this effect simply by thinking through the expected effect of differential growth. So if we have some disc-like structure, which is growing more rapidly in the center than at the periphery, we expect that in order to make this extra area available to growing and proliferating cells in the middle, it's going to buckle into vaguely hemispherical structure, whereas if it's overgrowing at the periphery, we'll have to make more distal area, and that will have to sort of curve out into kind of negative Gaussian curvature structure as here. So we understand this well mathematically, and physicists and engineers have used it as inspiration for sort of shaping elastomeric sheets by sort of baking in differential expansion and then designing and generating different three-dimensional shapes. So we can start thinking about how you encode a shape of a sheet that can generate intrinsics of internal area from within, as you know, a plant leaf can do by non-uniform growth. Well, so we know basically how this, you know, the basics of how it works from Gauss long ago. We basically told us that any 3D shape with topology of a disk in 3 dimensions can be sort of isotropically flattened into a disk. So it is conformally flat. And conversely, therefore, we can generate so any 3D shape by non-uniform isotropic growth in the disk. When we're talking about shaping of these sheets, we were already talking about mechanics, right? Because the whole business of buckling and accommodating extra cells by generating a surface with more area, it's all about mechanics. And there's clearly a very obvious role of tissue mechanics in defining the shape part of morphogenesis. But control of growth is not the only is not the only place where mechanical behavior enters. Let's get away from disks and talk about spheres, or rather ellipsoids, which is what a fly embryo is like. So we're going to look early in fly embryonic development. And here you're looking at light sheet microscopy. I think this is sort of still cut off because there should be a picture of Sebastian Shraiman there, who is light sheet imaging I'm presenting here. Anyway, so you're looking at the nuclei lined up on the surface of the embryo. And of course, all the nuclei are genetically identical, but at this stage, some of them have decided to express different genes. And the yellow refers to one particular type of early developmental transcription factors. So there is pattern formation occurring on the level of gene expression. But the next thing that you're going to see in this little movie is, so this is the ventral side of the embryo. This is actually the dorsal view. You're going to see cells on the ventral line dive in, and that's so the first step of gastrulation. And the next thing you're going to see is that the cells from vaguely this ventral area will rush over the posterior pole onto the dorsal side. So there'll be sort of a major migration of cells. So I guess here we start from the beginning. This pattern of genes kicks in, and then you see this gastrulation and what's called convergent extension, germ band extension flow. So the major rearrangement of cells on the surface of the embryo. So Sebastian, and now you see his picture, likes unrolling this embryo onto kind of a cylinder, this sort of Mercator projection of the embryo, and then you see a little better which cells are going where. And he also does sort of image flow velocimetry. And we'll map out this whole flow for you. And here you're looking at one particular snapshot, but he has the whole movie of it. And the general structure of the flow is quite simple. It is it is locally hyperbolic flow. So you have right. So this is sort of a cylinder which got cut up along the ventral midline and unrolled, right? And you have these two hyperbolic fixed points. Cells are moving towards the ventral side, if you like, towards the belly button, which it doesn't have, and elongate along anterior posterior axis, right? And if you have a flow like this on the ventral side, then on the dorsal side, you basically have to have the opposite flow. You have, again, this hyperbolic flow in with opposite orientation. And for topological reasons, you have to have 4 recirculation zones to straighten everything out. So there is something that any physicist with background in fluid mechanics will love. This is a flow. We can study this flow of tissue, but there is an immediate question. So these cells are flowing, but is animal epithelial tissue, is it a fluid or is it a solid? And it clearly isn't your undergraduate or even graduate continuum mechanics, because this is cursed here. Because cells obviously generate their own internal forces and have internal activity. And last but not least, tissue contains information, right? We saw these patterns of genes that were activated in some non-trivial patterns. So how do we deal with this? So I just showed imaging, which was based on nuclei, but you can also very nicely image cell interfaces, and then this epithelial tissue will look like a nice polygonal array of cells. And for a moment that we can look inside the cells. So what would we see? Well, we would basically see actomyosin cytoskeleton in this sort of very simplistic view of cell mechanics. Stress is concentrated in the cytoskeletal belts that live right below apical surface of the cell. And you all know about actin and myosin contractile filaments. And in this epithelial cytoskeletal cortex, they are welded together by cadherin. Okay, so we got our cytoskeletal cortex, and three different relevant molecules. Of course, in reality, there are hundreds and hundreds more. So the first question for a modeler dealing with this is, when do we stop? Just how complex a model should be. And we think that the simpler the better. So rather than add more layers of structure and control, we're going to strip molecules and replace them by sort of phenomenological description, which hopefully gets to the essence. And the essence for us is geometry. We want to describe the geometry of cells, and mechanics, i.e. the forces. And so the simplest description would basically attribute some tension, in general, actively generated tensions associated with cytoskeleton at the edges. And whereas tension, there has to be some pressure to prevent cells from collapsing. So we'll say that there is some pressure associated with each cell. And we'll just say that these forces are in balance. So tissue is instantaneously in mechanical equilibrium. And mechanical equilibrium basically means, okay, I'm going to simplify things a little bit. Let's approximately assume that pressure differentials between the cells are small compared to tensions. And then force balance just means that tensions acting on a given vertex add up to zero, right? So we have 3 vectors that add up to 0, which basically means that we can rotate them and build a little triangle. We can represent local force balance as a triangulation of a plane, right? Of course, this plane lives in tension space. Local force balance has a geometric implication that there has to be this hidden triangulation of some abstract plane describing this geometry. And that abstract plane has a very real consequence, because you can convince yourself by doing a little bit of high school geometry that the existence of a flat triangulation like this implies a particular relation between some angles that are observable in polygonal tiling. So it's basically sine law on steroids. The ratios of sines computed around the plaquette has to be equal to 1. So the only thing that goes into this is the assumption of a force balance. We don't need to know anything about myosin or actin or who is generating forces, only that there are forces and these forces are localized cell interfaces. The rest is geometry. But we can immediately test that. And by testing it, we then test our hypothesis that this tissue is at least in the approximate force balance, and these forces are localized at interfaces. I guess I should have said it in opposite order. The forces are localized at interfaces and they're balanced. But we have these nice pictures of tissues, and all we have to do is look at the angles and take these measurements very seriously, and we can then of course, everything is approximate, and the measurement of these angles is also approximate. So we don't expect to find 1 for every cell, but perhaps something close to 1, and close to 1 in the sense of statistical significance. So we want to find something that's much closer to 1 than expected in some suitable random ensemble. Yes? Yes, absolutely. Yes, you can. Not here, but you absolutely can. And you can also take account of bending edges. And it's actually quite elegant. Anyway, so the bottom line is you find this overrepresentation in some tissues, but not all, right? It's not obvious that this tissue satisfies, so given epithelium satisfies the assumptions that we're making, but there are some that do, and that's what we want to look at, right? But now a little bit encouraged by the simple assumption being reasonable, we move on and build a little further. So, I just argued that for this polygonal tiling in mechanical equilibrium, there is a triangulation. But you can ask the opposite question of, given a triangulation, does it specify a unique arrangement of cells? And it turns out that it actually doesn't. There is a whole family of cell shapes that have identical force balance triangulation. And these different cell shapes are related to each other by a deformation that does not change angles. So we call it an isogonal deformation. Well, so having these sort of soft modes is something that you often encounter in physics, and it has implications. When there are these soft modes, it means that you expect to see them as sort of common and large fluctuations in reality. So we can go back to the data, and in this particular case, look at deformation of cells, just before the ventral furrow formation. There are cells undergoing major deformation of shapes, and you can then ask, so look carefully at what the angles are doing during that deformation, and you can convince yourself that sort of, well, I guess 80% of variance in that deformation can be attributed to this isogonal modes. So you actually see the propensity towards these isogonal deformations in real tissue. So let me see how badly I'm doing in time. It's terrible. Okay. So I hope I convinced you that 2D geometry of cells is constrained by mechanical equilibrium. So in fact, this constraint is so strong that you can use it in reverse by sort of formulating kind of a mechanical inverse problem, where by analyzing so cell geometry, you can try to infer tensions and pressures in reverse. And okay, so you can do this analysis, and then the question is, how do we test it? It's very nice to be able to deduce tensions in the cell network, because they're very hard to measure directly. To sort of see it, people typically use ablation and recoil. That's a good way of visualizing and trying to quantify tensions, but by definition, that's a destructive measurement. So what else can we do to test in sort of higher throughput? Well, let's make seemingly plausible assumption that, okay, these tensions are generated by myosin, so maybe if we can visualize myosin and look at the relative abundance of myosin on different edges, we can say something about relative tension. And this particular data comes from Thomas Lequi's lab, and Madhav Mani, when he was a postdoc at KITP, actually a long time ago, carried out this analysis. And what he did was he actually did the analysis in essentially on cell scale, and correlated predictions from this mechanical inverse to this myosin signal, and plotted the distribution of correlations. And he basically found that the median correlation was 0.4. And it was so hugely statistically significant compared to the control, where you scramble the geometry and where obviously correlation is 0, medium. But it leaves us with a sort of peculiar situation. Do you think that correlation coefficient 0.4, is that a lot, or is it not so good? Well, I would like to argue that it's actually very surprising that there is any correlation at all, because we were just analyzing this tissue as if it is static, right? Mechanical equilibrium. But in reality, this is actually just a frame in the movie. And you get decent correlation frame by frame. And now actually something very peculiar is happening. So we have this fluid tissue, which instantaneously is behaving as if it is in force balance, approximately. But now we can so start thinking of this relatively low correlation coefficient as coming from two different sources. One is, of course, all the horrible approximations that had to be made along the way. But the other one is so the fact that force balance is only approximate. So again, it's approximate, but still useful. So now we have to move from this static picture to dynamic picture. We started out very agnostic about where this tension was generated from. It was just tension. But now let's think of it more as kind of a Hookean spring, but a very peculiar Hookean spring with a little devil inside that is capable of changing the intrinsic length of the spring, right? And of course, that very active devil is myosin. So, and we know that myosin activity can contract actomyosin filament if it can, and that is controlled by how much external load is applied to the filament. So if there is too much external load, myosin won't be able to sustain it, and it will elongate. And if there is not a lot of load and too much myosin, then it will contract. So there is this dynamics controlled by myosin, but effectively now, if you coarse grain and just look at the behavior of the tissue that on whatever subcellular scale is defined by this sort of behavior, you expect to find effectively viscoelastic response, or I should say active viscoelastic behavior on large scales. But viscoelastic behavior on large scales means that on large scales, the tissue would be ultimately viscous fluid. But so here's my arm, and there is skin, epithelium, and I shake it, and it doesn't drip. It is not fluid. So what's going on? So how can we actually guarantee that so the solid behavior on longest time scales? So how can we get these local active actomyosin filaments to coordinate their activity so that they reach force balance, right? So we need to get this tension triangulation to balance forces, but now we say each filament is going to be at stall, right? Will be static only if it has the right myosin level. So we need to coordinate how much myosin is active on each edge here. And how do we achieve that? Well, you do this, of course, with some suitable feedback. But what would be the feedback? So you can have negative feedback where or positive feedback where myosin recruitment is dependent on tension itself. And that wouldn't do it. However, if you make myosin recruitment dependent on the rate of strain, that would do it. And the intuition is very simple. So rate of strain, so if you have this interface with too much tension, right, more tension than the myosin can support, it will elongate. So And this rate of elongation then becomes a signal. Please send myosin, right? On the other hand, if it is contracting, that's a signal, get rid of some myosin. And if you have a feedback like that, you can basically guarantee that this array of cells will stably reach mechanical equilibrium. And that's a little model prediction. And it's really just the simplest way that you can think. Well, Sebastian Shraiman and his students actually went on to test this by doing experiments, both observing in fly lateral ectoderm and doing sort of active perturbations, sort of doing optogenetics. And lo and behold, they can measure this sort of dependence and in fact nail the prefactor here, which is basically over the 2. Of course, this isn't just a linear law, but the effect is nevertheless there, as one could have, well, as hoped for. Okay, so where does this leave us? It leaves us with the situation with a model that describes tissue mechanics on 3 different time scales. So fastest time scale is essentially elastic response. Intermediate time scale is remodeling of these active cytoskeletal springs by recruiting, I'm sorry, by contracting or extending. And together, they, as I argued, would result in a viscoelastic behavior. But then in addition, there is yet slower dynamics of actually recruiting myosin to interfaces. And just a moment ago, of course, I was focusing on mechanical equilibration, on reaching equilibrium in a network like that. But to understand this conversion extension flow in embryo, we also need to generate the flow. So, but we can very easily imagine some positive feedback, which is controlled now by patterns of gene expression of the embryo. But positive feedback to tension, which can then destabilize tissue and generate the flow. But still, our emergent picture is that tissue flow is better understood as remodeling of internal force balance, right? And I'll give you a real hand-waving argument. So, here is my hand, and I'm waving it. And it's moving, but it's not moving ballistically. It is really moving while being in force balance, right? My muscles are balancing the gravity and keeping all the torques balanced. And then I'm just remodeling this balance any which way I want. So it is happening all in, so adiabatically in approximate mechanical equilibrium. So that's the way we want to think about the tissue. Okay. So let me look at the clock. So Yeah, I can basically talk for another hour, but I won't. So I'm just going to fast forward to some conclusion. So that's the way we want to think about it. But then, of course, this is just a model, and there is a lot that we need to test, and you know, right? Models should explain and predict, right? And we really want to do both. So, you know, we can go back to Sebastian's wonderful measurements and see if we can explain observed flow. Well, he can also measure myosin distribution in over the whole embryo, and what's important is so amount of myosin, but also myosin anisotropy. So we actually have to describe it not with one number locally, but with three numbers. And if we have these three numbers, we can basically think of this as a proxy for local actively generated stress. And I said that on some intermediate time scale, the model is basically viscoelasticity. So if you know stress tensor, you can solve some equations and compute the velocity in terms of so a small number of parameters. And the bottom line is that you get, again, I would say a surprisingly decent agreement between observed and predicted flow. So I guess the question is, what was observed, what is predicted? Predicted is blue, observed is red, and these are so the errors. And right, these are non-trivial, in fact, spatiotemporal profiles, which one fits with a very small number of parameters. And it's actually pretty surprising that one can get a reasonable fit, to the extent that looking at where the errors are is actually informative of so both underlying biology and the model. But you know, so I just bragged about prediction, but there is no prediction here, in the sense that we still need to know, right? We're just relating these myosin observations to observed flow. But what we really would like to know is we like to predict the future. You know, can we predict what's going to happen in the next moment? And for that, we need to understand what drives dynamics of myosin. And to do that, one actually needs description on the cellular scale. And this is sort of the most recent piece of work driven by Fritjof-Browns at KTP, a postdoc, and Nicholas Clausen, a physics student. And they did sort of much more detailed analysis, looking at sort of beautiful, so fully segmented tracked data on the embryo that we got from originally also Sebastian Shraiman, and analyzed by Thomas Stern at Princeton. I'm way out of time, so I'm just going to give you the highlights here, in the sense that what we learned is that it is actually useful. I'm sorry. Yeah, what did we learn? So Fritjof and Nicholas can actually build a model of myosin dynamics entirely in terms of sort of positive and negative feedback on myosin, which is modulated by large scale on large scale by presumably gene expression, but also curiously depends on large scale morphology of the tissue. It turns out that it is important that the embryo is an ellipsoid and can support sort of balanced stress profile, sort of a hot dog stress. Hot dog always bursts azimutally because the stress is, azimuthal stress is higher than the longitudinal one. So there is already, if you like, this mechanical coordinate system available to the cells, which then appears to organize recruitment of myosin. So there are various predictions that I'd love to tell you about, but not tested yet. Well, I guess the overall message is that what I presented were sort of very, very simple models, kind of minimal models. We really we're trying to get away with this is my sort of philosophical interlude here again. We're trying to get away with minimal assumptions, like force balance. We believe force balance, and it doesn't matter where forces are coming from. But I find it really surprising that these simple models can actually predict something about biology. So, we think of sort of the simplest way to reach mechanical equilibrium, and then nature somehow obliges us by using this simplest method. I find this very encouraging and exciting. So maybe biology, so our ultimate complex system, is simpler than we think. And this is definitely something you can ask me about in the pub. So what happened to all the complexity? I will conclude by acknowledging again my friends and colleagues and collaborators that I pretty much already mentioned. So Fritjof Franz and Nicholas Clausen are responsible for that last monumental analysis that just got posted on the archive in the form of two preprints, one 50 pages long, another one I think is 30. So one is sort of empirical paper, another one model building paper. And the earlier story about the active tension network is the work done together with Nicholas Knoll, Madhav Mani, Himskirk. I talked about Dylan's thesis work on this anisotropic growth. I have acknowledged Sebastian's many, many contributions, all the imaging and analysis that he has done. And we've enjoyed and continue to enjoy wonderful collaboration with Eric Wischhaus and Matt Lefebvre. Thank you very much.